Right. Well, thank you, Fabrice and Dr. Solari and ARC for the kind invitation. I really appreciate it. Now, I will preface by saying I've really enjoyed the talks that I heard today from everyone, both the preclinical and the clinical sessions. Well, I'm giving a very unusual talk. I've actually never given a talk like this before. Typically, if Fabrice invited me to speak at um, at Gustav Rusi, I would be speaking on all the mouse models we have in our lab, or the single cell data that we are generating from our patients. But I don't think that was the intent of the invitation. And I think what Dr. Solari and others wanted me to speak about was sort of strategy, scientific questions, what is important as the foundation pivots towards a substantial investment in pancreatic cancer research in France and beyond. So that's why this talk is not going to be speaking much about primary data. In fact, much of the science, or actually the very little science I'm going to speak about today, is actually other people's science, and is things that have been generated in teams that I have led over the last decade or more. And again, just to emphasize that my talk will really talk about what are the burning questions in pancreatic cancer, and what are the sort of the major areas, and how do we sort of pivot to them in a meaningful way. So this is the building I'm in. This is where the PANCREAS Center is located at MD Anderson. All right. So again, everyone knows about the rising burden of pancreatic cancer deaths. These are data from the American Cancer Society for last year. Pancreatic cancer is the third most common cause of cancer-related deaths. Its rates are going up, and in the next decade or so, it's going to overtake both colorectal and breast cancer, and be the second most common cause of cancer-related deaths just after lung. In the state of Texas, where I'm at, you can see that both the cases and the deaths keep going up every year. So there was this very nice article, "Why is pancreatic cancer on the rise?" in Scientific American, and the reasons are many. It's an aging population. People are living longer, which is fantastic news, but unfortunately, it also means that we are going to get more pancreatic cancer, which is a disease of the elderly. We are better with the diagnostics, so we can now say when something is pancreatic cancer versus, disseminated metastases without any origin. And of course, diabetes and obesity have a very important role to play in terms of increasing the risk for this disease, and so that's another factor that's contributing. And in fact, as Dr. Wolf, who's one of our senior oncologists at MD Anderson, who was quoted in this article, said, the profile of patients with pancreatic cancer has changed. I'm sure many of the oncologists on this meeting today have also seen this. Many patients are now obese or hypertensive, and some of them smoke. So all of which are risk factors for pancreatic cancer. And
Anirban,
yes.
Can I introduce you? Which slide are we supposed to see? Because I'm still on the slide with the very nice building that we would all love to have, but I assume you are talking about another slide.
Oh my gosh! I'm so sorry.
I think it's us to.
Are the slides not moving? Okay, maybe is this not? Is it moving now? Can you see it?
Yes.
Can you see it now?
Yes.
Yes.
Okay, great. Thank you. Fantastic. Okay, so I was just talking about this slide, about this article, and the fact that there is no, in most cases of pancreatic cancer, there is no one single attributable risk factor like you have with HPV-related cervical cancer or lung cancer with smoking. In most cases of pancreatic cancer, except that small subset that arises in the backdrop of germline mutations, we don't really have a single attributable cause. So the last session that just finished was on therapies in pancreatic cancer, and it was really very interesting to hear. But the fact of the matter is, from the time that gemcitabine was first introduced to now, when we have these two first-line regimens, there hasn't been a lot of progress in terms of survival for advanced patients. We certainly have seen significant advances in the adjuvant setting. In fact, the only patients who truly have long-term cures in this disease are the ones who are lucky enough to be operated. And in fact, much of that adjuvant data has come from France. So I think, but in terms of advanced metastatic disease, we still struggle with the regimens. We have 2 first-line regimens. We have a second-line regimen. There are some comments in French. Is it okay? You can see the slides. I don't understand what the comment is. I just want to make sure you can see the slides.
Maybe Anirban, I'm going to tell. I think it's everyone to synchronize with you if we want to see your slide. So you're not supposed to make any action. I think it's us to synchronize with you with one button. So
are you all able to see the slide where I'm talking about therapeutic challenge of pancreatic cancer?
Yes.
Yes.
If we are synchronized, yes.
Okay, good. Perfect. So I'll keep going. Okay. All right. So again, coming back to sort of the intent of my talk, which was really to talk about scientific questions and strategy. And again, this is obviously a very, very challenging disease. And in order to make progress, unfortunately, we can't just leave things to wishful thinking and some powerful jargon, as this cartoon says. So in the last decade or so, as I said, I've been involved in many of these sort of strategy sessions or deciding what the high priorities for research should be. And that has been at the national level with foundations such as ARC, as well as in my own institution with the Moonshot. I'm going to talk a little bit about all of them because I think it's important to sort of say how each of them is different. And they are different. What happens in the national level, which has significantly larger amount of funding, is very different from what happens in a foundation and very different from what happens in the institution. So the first effort to sort of address progress in pancreatic cancer was in 1999. Oh, just something happened. Are you able to still see the screen?
Your slide. No, not anymore. We can't see your slides anymore.
I don't know what happened. Hang on. Suddenly it kicked me out. This file already exists. Replace, upload, replace, maybe. I don't know what's going on. So if this has a problem, maybe Daphne, I sent you Delphine, I sent you the slides. Maybe you can show it. If I have a continuing issue, you can show it. Let's see.
Yes.
Teams is well known though to create problems.
Maybe Anirban, you can send if it's good.
Yeah, no, I've already sent it. If there's a problem, you know, I'll just have Delphine maybe drive it from there, and I'll just speak. It's fine, not a problem. Can you see it? Hang on. I apologize for this, guys. Okay. All right. Okay. So the first concerted effort by NCI to address progress in pancreatic cancer actually took place in 1999, and this was in Park City, Utah. Oh, it keeps doing these funny things. Hang on.
I'm sorry. We also have some technical issues here.
Okay.
Okay, let me know if it's okay for you. I don't know, maybe I'm not on the right slide.
If you can go one slide back.
One slide back.
Yeah, right here, this here. So should I drive or are you planning on driving? Do you want me to drive? Yeah. Okay. So what do I do?
You can ask.
Do I just go one slide?
You can ask for control, I think.
Oh, okay. All right. Why don't you drive? Maybe it's easiest that way. All right. So next, if you wouldn't mind, next. Click next, please. Thank you. So this was a white paper that came out of that meeting. So this was the first time that the NCI said, we need to make progress in pancreatic cancer. There isn't enough progress. Let's get a group of people together. This was in 1999. And this was the paper that came out of that meeting. Okay. And this table in that table 5 was the main research opportunities that at that time the NCI and the group of scientists felt were important in pancreatic cancer. Now almost more than 20 years ago. Next click, please. And so there were several things that were obviously noted in this table, but two things that in particular rose to the top and which actually became reality very soon after were the development of relevant animal models and mapping of premalignant lesions in the pancreas, so PANINs and cystic lesions. And again, I'm showing these examples to tell the foundation that you need to make this sort of a strategy, come up with the high priorities, and then act on it, because otherwise, if you don't have this sort of directionality, there are many, many interesting things we could do, but you have to prioritize what you can do in a meaningful time. So next slide. So as I said, this happened in 1999. Within 3 years, David Tuvison had developed and Sunil Hingorani had developed a mouse model. We actually were collaborators on that paper, and many of you now use these KPC models. I actually saw data today from several of the models that use this basic backbone. And then we described the whole PANIN progression, the histology, the genetics, etc. So again, this would not have happened unless we were sort of focused on doing this based on the Utah think tank. Next, please. And then subsequently, Bert Vogelstein and colleagues at Johns Hopkins did the first exome sequencing of pancreatic cancer. This was in 2008, and it's hard to believe. It's not that long ago. 2008 was only, I don't know, 13 years ago, but this was all done by PCR, by hand, thousands of PCRs, graduate students and technicians running these PCR reactions every day. And now you probably heard from Andrew Biankin this morning, if you can click the next slide. Now we have all of these large data sets like the ICGC and TCGA, and everything is done by next generation sequencing. There was a lot of data that speakers presented today on sequencing, on TMB, on sequencing organoids, single cell. So we've come a long way in terms of capabilities in just a few years. But again, this was not that long ago, and now we know a lot about pancreatic cancer, including the big player in this disease, which is KRAS, which is right there on the top of this circle. Next slide, please. So that was 1999, and then for 15 years, nothing happened. Science kind of moved on its own. The mouse models were made, the sequencing was going on, and new therapies were coming. And then in 2014, the NCI said, wait a minute, 15 years has passed since Utah. We need to get together again and see what are the major gaps. Where do we need to prioritize our research so that we can make progress against this disease? And so I was actually at this meeting in 2014, and we came up with 4 scientific initiatives that we felt were very, very important in pancreatic cancer. And I'm not going to read them, but I'll just say the first 2 of those 4 were basically focused on early detection. Because again, a lot had been done in sequencing, there was all of this work going on in therapy, but what we realized is that early detection probably has the highest chance of making an impact on survival in pancreatic cancer. Right now, only about 15 or 20% of patients undergo surgery, can get to our surgeons because they have resectable or borderline resectable disease. If we can up that number to 50% or 70%, the difference we're going to make on survival of pancreatic cancer is going to be better than any CAR T cell or whatever people are doing. And again, this is not to say that the therapies are not important, but early detection is something that clearly rose to the top for us. And then we also talked about immunotherapy, and in fact, several talks today were on immunotherapy and the challenges of having successful immunotherapy in pancreatic cancer. And this was again identified as a very important priority area. And the fourth one, again, you cannot do pancreatic cancer without RAS. RAS is everywhere. It's what starts pancreatic cancer. It's what keeps pancreatic cancer going. RAS is not just in the cancer cell. It's in the surrounding microenvironment. It talks to the immune cells. It talks to the fibroblasts. So to tackle pancreatic cancer, you have to tackle RAS. So these were our 4 scientific initiatives. And for the first time, the NCI said, typically, as you well know, the NCI funds individual investigators with these big, not big, actually medium-sized grants called R01 grants. But for the first time, the NCI said, we need to fund team science in pancreatic cancer. We need to fund big consortia because progress in this disease is not going to come from just funding single investigators. We need big teams. Next slide, please. And so I'm going to talk a couple of the examples that came out of this 2014 workshop. And again, these workshops are not just for the sake of having these workshops. They really help because they help the NCI prioritize and say, we need to fund this because the people who are working in the field, the experts said, this is an area that needs a lot of investment. And it helps us, the scientists who are in the field, because we say, okay, the NCI or whatever wants this to be a high priority area, so we should pivot our research here and write grants or try to study this because this is clearly important. So for example, I said the first two of the 4 bullet points were on early detection. And so, and again, the NCI really wanted to fund these big teams. So one of the things that came out of this meeting was this consortia, if you can click the next slide, this consortia called Consortia for the Study of Chronic Pancreatitis, Diabetes, and Pancreatic Cancer, or CPDPC, which is a big name. But the NCI has poured a lot of money into this because they think this is a very high priority area. And what is this all about? If you can go to the next slide. So this is this consortia that has now been established and which is basically recruiting about 10,000 patients, 10,000 individuals with new onset hyperglycemia or diabetes. So why are we recruiting 10,000 patients with new onset hyperglycemia or diabetes? What has that got to do with pancreatic cancer? Next slide, please. So work that was done by one of our colleagues from Mayo Clinic, he's now actually at MD Anderson, showed that many patients with pancreatic cancer have worsening hyperglycemia, including frank diabetes, several months prior to their clinical presentation with jaundice and weight loss, etc. If you actually track these blood sugar levels as early as 24 months to 36 months prior, you can start to see this worsening hyperglycemia, which meant that if you could somehow use hyperglycemia to enrich for patients who might be harboring an occult pancreatic cancer that has still not manifested itself, you might be able to identify these cancers earlier before they become metastatic and then get them to our surgeons so they can be resected. Again, this diabetes is not a risk factor for pancreatic cancer. This diabetes is caused by the pancreatic cancer that is still there. It's still tiny. It's not manifested itself, but it's causing the insulin resistance that's leading to this hyperglycemia. The problem is only about 1% of patients with new onset diabetes, actually less than 1% actually have pancreatic cancer. The vast majority is still regular new onset diabetes, type 2 diabetes. So how do we differentiate that 1% from everyone else? So that's what this consortium is doing. It's going to recruit 10,000 individuals. Next slide, please. We actually have 22 sites that are recruiting these. It says 8,000, but that actually their number should be 10,000. And we'll follow them for 3 years, and then we'll identify the percentage that will develop pancreatic cancer. We'll have blood samples from them, and we'll go back and see what is different in their blood samples versus everyone else that has that does not have pancreatic cancer. And again, this talk is not about new onset diabetes or anything. This talk is just to highlight we had that workshop in 2014. We identified a high priority area in early detection, and the NCI poured a lot of money into that, and therefore this cohort has now happened. In fact, I'm actually leading this cohort and the recruitment because again, this could provide an opportunity for early detection in pancreatic cancer. Next, please. The other area, as you know, I talked about immunotherapy was the third bullet of the 4. And so once again, based on our recommendation that immunotherapy and challenges of immunotherapy need to be addressed head on in pancreatic cancer, the NCI created something called the Pancreatic Cancer Microenvironment Network. Next, please. It's right there on the slide. And the goal of this network, which actually I'm the PI of the coordinating center, we call it the Pac-Man network, like the video game Pac-Man, is to identify, and these topics were discussed by several investigators today, so it's not anything new to many of you. But again, the idea was not to do that in single labs, but to put together a team of scientists across the country who would understand what the barriers to immunotherapy are and how can we overcome it. Next slide, please. And this is the Pac-Man network. You can see we have labs all over the country, on the West Coast, East Coast, in the middle of the country. And that's our little logo right there. And again, the goal of this is to come together as a group and do team science on overcoming barriers to immunotherapy. Next slide, please. So we had 1999, we had 2014, and then 5 years after 2014, in 2019, the NCI said, and again, this is very important. If you're going to invest in a disease, it's not just enough to say, here's the money, do what you want, thank you very much. You have to come back and say, well, what did you do? And do we need to do more? What are the still unexplored areas? What needs to continue, etc. And so 5 years later, they got us all back into NCI again, and same people, the same 25 people who were there in 2014, and said, now tell us what needs to be done. I don't know what happened on this slide. There's a lot of funny text, but anyway, the bottom line is we all went back to the NCI 5 years later to review what the progress was. Next slide, please. So I'm not going to go through every single line of text here because there's too much and there's not enough time. These slides are available to the ARC, and you're welcome to read through them and study them, and whatever questions you have, please reach out to me later. But again, we divided the main sort of burning areas that we need to refocus in pancreas cancer into 4. The first was tumor biology. Again, you have to study the tumor, and that's why you had so many talks today on tumor biology. You see the big KRAS is right there, and the K is in brackets because there are other RASs in pancreas as well, NRAS and HRAS, but K is the most important. You got to understand KRAS. If you click the next slide, please. This time we had a few additional things that were not in 2014 in the priorities that we added in the tumor biology area. And I want to talk about 3 of them because they're very important in pancreas cancer. And it's interesting because some of you just spoke about this in the last session. And the oncologists on the call on this meeting today, you will really appreciate the pathophysiology of cachexia and sarcopenia. Muscle wasting and cachexia is incredibly important in pancreas cancer. Our patients, their survival often is impacted by this. And so this is really understudied in pancreas cancer. And so we felt it was important enough that it should be a line item in the priorities for study in the next year. And in fact, there are now grants. There's a cachexia related network that has been formed to particularly study cachexia, and not just in pancreas, but it happened because we talked about this in the context of pancreas. Role of the microbiome, the talk we just had talked about this. This is incredibly important. In 2014, we didn't talk about the microbiome, but then all of this wonderful work happened, including from France. And so now we have microbiome as an important line item in the priorities. And then this last point, and I take some credit for putting in there because I think this is very important. As you know, I'm a pathologist by training, and I love mouse models. I have a lot of mouse models in my lab. There's so much we can learn from mouse models. But ultimately, pancreatic cancer science has to happen in patients. We have to learn from our patients, and that involves doing longitudinal biopsies, pre, post, on progression, trying to figure out what's happening in vivo in our patients, what's changing in the microenvironment, what's changing in the tumor cells. Without that science in patients, I just don't think we can learn fully. We can learn a lot from our mouse models, but not fully unless we do these correlative biopsies. Next slide, please. So tumor biology was the first one. The second one was animal models. Again, you have to have animal models. They are not enough by themselves, but they absolutely need to be there. If you can just click the next slide. And again, we had the original KRAS and KPC models, but what those models didn't incorporate, but I think it's very important in the context of pancreatic cancer in our patients, is the risk factors that we know that our patients have: diabetes, obesity, pancreatitis. So can we build models that encapsulate these risk factors and also the RAS mutation and the P53, etc.? And then models that facilitate immunotherapy. As you well know, many of you are expert immunologists on this call more than me, that there are mouse and human differences, and that impact the lessons that we learn and sometimes can lead us astray. And so it's very important that we build models that reflect what's going on in humans, including, for example, humanized mouse models, where the T cells and the ligands and the cytokines, everything is human, so that it better reflects what's going on in our patients. Next slide, please. You cannot do pancreatic cancer without prevention, screening, and early detection. I mean, I've already talked about that. In 2014, 2 of the 4 bullet points were early detection, and of course, we retained a lot of that. If you can click the next slide. One thing that is particularly important in the US that we know is that many of our studies in pancreatic cancer, including genetics and biomarkers, are heavily biased towards white populations. We need to study our African-American citizens and Hispanic citizens. Many of them don't participate in clinical trials. So one particular area that we emphasize and that the NCI has completely co-opted is the importance of minority representation in these studies. Next. And treatment, of course, you have to have treatment. And again, the top bullet is RAS. And this is the big player in pancreas. If we can target RAS, there's a lot we can do in terms of pancreas cancer. And then there are other bullet points. Again, these slides are available. These are all public bullet points also on the NCI website. So you're welcome to look at them. Again, I just wanted to highlight this tissue acquisition science in patients. We have to do our science in patients, accompanying what's happening in the lab, whether it's in organoid models or in PDXs or in genetic and engineered mouse models. And in fact, we have to keep going back and forth. What we learn from the science in patients, we have to model back in our mice, and what we learn are organoids, and what we learn there, we have to take back in terms of the next clinical trial that we are designing. Next slide, please. So all of that helped. I mean, all these meetings were not in vain in 1999, 2014, 2019, if you can click the next. So in 1999, when the Park City, Utah Summit was held, the NCI only contributed $17 million or so to pancreatic cancer. Now it's over it's over $200 million. So it's been a huge increase every year in terms of pancreatic cancer investment, and it has happened because those strategy sessions happened, the priorities were identified, and the NCI responded to those priorities to say, we are going to fund early detection, we are going to fund KRAS, we are going to fund immunotherapy consortia. Next, please. The other thing that has happened is now, I don't know how it is in France, but we also have the US military has a medical research unit called DoD funding for cancer. And for the first time last year, they decided to start funding pancreatic cancer research. So, and in fact, it's a small amount compared to the NCI, $15 million this year, but it's a start. And again, if you can click the next. So I'm actually the chair of the program committee for that sets the priority areas for this. And one of the things we particularly emphasized, and I'll come back to this in the last slide, is these partnership grants between a senior PI and a junior faculty, so that again, we encourage team science, that this doesn't go to just the senior professors only, who do their own stuff, but we really collaborate between someone senior and someone junior, so the junior person gets mentored and is able to do great team science. Next slide, please. So that was all the NCI. This is the big player in the national scene. As you saw, they have a $200 million budget for pancreatic cancer, but there's also foundations. And so what are the foundations and what are they doing? So in the US, there are mainly 3 foundations funding pancreatic cancer research. There's a Lustgarten Foundation, there is Pancreatic Cancer Action Network, or PanCAN, and there is Stand Up to Cancer. And these are the 3 websites. Next, please. So I'm going to give a couple of examples. Again, with foundations, unlike the NCI, which has a much larger budget, with foundations, you have to prioritize. And so Lustgarten Foundation actually is the largest private funder of pancreatic cancer research in the country. And they have decided to do 2 things. One is they're going to fund some of their own research, and 2, they decided to partner with Stand Up to Cancer, which is the other foundation, so that together they can fund a lot of different team science efforts. So Lustgarten's own effort is something called the Clinical Accelerator Initiative. I'm actually a part of this, and the goal of this CAI, which is run by this thing called the Translational Advisory Group, is, if you can click the next slide. So something really exciting is published in a preclinical model. Boom, we put together a clinical trial, we fund it. We try to get the drugs as soon as possible from the company so that the translation, that's exactly what the name suggests, translation, clinical accelerator initiative. Something that's really exciting, like this cancer cell paper on the CD155 TIGIT axis from MIT was published earlier this year. Even as this was happening, we got a proposal from the group and at Lustgarten, and now this is going to be funded as a clinical trial targeting TIGIT in pancreatic cancer. So the idea is something exciting on the horizon that's happening that is really fascinating preclinical science. Boom, we get the clinical trial done, because ultimately that's what's going to matter. Does it work in our patients? Next slide, please. This is the Stand Up to Cancer, and Stand Up to Cancer, as I said, what they have done is something very clever. They have partnered with Lustgarten, so basically now you get more bang for your buck, as they say, and they are funding a lot of different teams. This partnership together is called the Pancreatic Cancer Collective between Lustgarten and Stand Up to Cancer, and there are many teams that are funded. If you can click the next slide. And the goals of this, again, very important part about team science, and I'll come back to this. If you read the first bullet point, inspire collaboration among people who haven't worked together. This is a very important aspect of making progress in a disease like pancreas cancer. You can't just keep funding the same people who have worked in pancreas cancer for 20 years. You have to bring in new people, including physicists, engineers, data scientists, people who may have made, prostate cancer models or whatever. If they have a great idea that works in pancreas cancer, well, get them into the door and fund them, because that's the only way we're going to make progress. If you can click the next slide, several teams have been funded, including us. We are funded for a team on cancer interception. Nuria, I think she's on the call. She has been funded by this to develop computational approaches with Raul Rabadan at Columbia for identifying high-risk pancreatic cancer population. So their grants are not just restricted to the US, they also fund folks in Europe. So again, a lot of these teams are funded, and again, the idea is to bring together people who may not necessarily work together, bring together expertise that may not be necessarily considered pancreas cancer specific, but which can make a difference in terms of moving forward. Next, please. So I'm going to talk very quickly about our team that got funded by Stand Up to Cancer. Again, the idea is not to talk about the science too much in detail, but just to sort of tell you the kinds of things that are funded as part of these team science efforts. So the goal, overarching goal, and this is our team, this is a pretty big team, and it includes MD Anderson, Johns Hopkins, San Diego, Dana-Farber, Mayo Clinic, MIT. The goal of this was to identify high-risk cohorts in pancreatic cancer, and then do something about their high risk in terms of preventing the progression to pancreas cancer. And that's why it's called interception. We are intercepting cancer from happening in individuals who are at high risk. Next slide, please. So 3 aims: enhance germline testing for cancer interception. So basically identify through germline testing who's at risk. Next, intercept pancreatic cancer using targeted immune prevention, and then blood-based markers. If you can click the next slide. We have a couple of different, next slide, a couple of different clinical trials as part of this. If you can go to the next slide. So this is the trial that was run through the Stand Up to Cancer team called Generate, which stands for Genetic Education, Risk Assessment, and Testing. And the one thing you will see about all of this, and this is very important in team science, is that there are leaders who are coming together, and there's not one person who's leading everything. In fact, even though I'm the overall lead of this team, my goal is literally to put the progress reports together and just present. The actual work is being done by designated leads who are experts in respective areas, and that's a big part of team science. Is that there are really sort of folks who are leading efforts within the big team. So this is being led by Dr. Sapna Singhal and Judy Garber at Dana-Farber, and what they did here was a very, very innovative trial where they actually used internet-based, completely online approaches to identify patients who are at risk for pancreas cancer, who get tested for germline mutations at their home. They never have to come to the hospital. A kit is sent to them. They spit into that kit, and then the kit is sent back. And then if they are positive, then they are entered into surveillance programs. So everything is done from their home. And during the pandemic, when we had so much disruption in our hospital system, the Generate study actually accrued very well. In fact, it accrued better than expected because patients loved being able to do this testing from their home. They didn't have to come out anywhere in the pandemic and go to a hospital. So next slide, please. And this is next slide. This is the Generate team. Well, if you can go back one slide, sorry. So just to show, this is the multiple sites. And again, this study is soon coming out. It's been accepted for publication, and we identified almost 100 patients with germline mutations who did not know that they were carrying a germline mutation, and now they have all been entered into various surveillance programs near their homes. So again, this is one of the aims of our study. Next slide, please. The second, and again, we have multiple aims. I just wanted to highlight a couple. This is an aim that's being led by Dr. Liz Jaffe, Nia Zaidi, and Mike Goggins at Johns Hopkins. So again, different institutions are leading different parts of our team. That's another very important tenet. Everything should not be centered with one person, one institution. You have to divide and conquer. So this is an immune interception using a RAS vaccine in these high-risk cohorts. So these are patients who have germline mutations, who have worrisome findings in their pancreas, not cancer. They haven't gone to cancer, but there are things happening in the pancreas that looks worrisome. Normally, many of them might get surgery, but here for the first time, we are using an immune interception, a vaccine-based approach, not to treat cancer, but to prevent cancer from happening. Next slide, please. And this is the design of the trial where we give a prime and then a boost, and these patients are followed. And again, the goal of this is a pilot study, so we are not looking necessarily for a clinical impact here in the time of this grant, but we are looking for a biological impact, which is, are we getting RAS-specific T cells in the periphery of these patients that we can measure, which would tell us that we are generating an immune response, kind of what people, I guess, are doing with SARS-CoV and vaccines, the same thing. Are you getting T cells against your antigen of interest? That's the main objective. Next slide, please. The third is this preclinical models. So we have the clinical part with Dr. Jaffe, and now this is MIT. So again, we had Dana-Farber, we had Johns Hopkins, this is MIT, and so this is work by Tyler Jackson and Wilfried Pastor. Next slide. And this was the paper I just talked about with the Lustgarten. They published this in mouse models. This was actually funded by our Stand Up to Cancer, and now through the Lustgarten clinical translator, clinical accelerator, this has now gone into clinical trials. So very rapid from the mouse model, published, and then into clinical trials. Next slide, please. So I've talked about the NCI. I talked about foundations using Lustgarten and Stand Up to Cancer as examples. And finally, now I'm going to talk about my own institution, MD Anderson, where we have the Moonshot effort. And I actually lead the Moonshot in pancreatic cancer. I've done this now for about 7-8 years now. And it's been very interesting. So I'm going to talk a little bit about what are our sort of general philosophy of the Moonshot, how we do it, and a couple of exemplary projects. So next slide, please. So why do we use the Moonshot mechanism? I think the most important thing is we use it to fund grants that the NCI would not fund because they're very high risk. They don't have a lot of preliminary data, but if they work out, there's a lot of reward that comes from it. The second thing is we actually use the Moonshot to develop indigenous assets because MD Anderson owns those assets then and can either license them or make a new company. And so part of the Moonshot is also strategic. We want to make it into a self-sustained venture, which sort of generates its own revenue. And the third thing that is very, very important in any initiative like this, and is a key component of team science that I'll come back to later, which is you need rigorous peer review. This kind of work cannot be done with favoritism. You can't do this in, 5 people get together over coffee and decide, well, this is how we're going to divide the money up. This has to be done in a very transparent manner before external peer reviewers who will basically say up or down very clearly. And so that's a key component in order to retain trust in terms of the progress that we are making. Next slide, please. And over the years, I just show this as an example, that we have gone, the moonshot has funded a lot of indigenous MD Anderson efforts that have gone from preclinical validation to clinical trials to being licensed to companies. Some of them, because they either didn't work or didn't look like they were doing as well as we were hoping, have been graduated. We call it graduated, but basically part of leading a team science effort is not just to be a happy messenger, but also to convey the bad news, which is, I'm sorry, but we have to go on without you because this is not working. We need to change this project. So I think that's also part of leading this team science efforts, is to not just always be the bearer of good news, but sometimes take hard decisions if you're not getting optimal progress. And that's what you see in some of those graduating hats. We call them graduated from the moonshot because they were not doing well. Now, sometimes they graduate because they get licensed by another company and so they're off the moonshot. But again, it's very important that we stay accountable to the progress that we said we are going to make. Next. So there are 3 models. Oh, again, just to highlight, we tell this to every investigator who comes on the moonshot. It's not an ATM. Don't expect to keep getting funded year after year after year. You're going to have to show progress, otherwise you're off the moonshot. So we have 3 models of how we run these projects over multiple years. Model 1, there'll be several clicks, so you can just go through several clicks and let's just get to the bottom of it. So if you can just click several times. Stop. Thank you. So this is a drug that is an OXPHOS inhibitor. So we supported previously through the PANCREAS Center the development of this drug. This resulted in a Nature paper from Julio Droeta. Then we funded through the moonshot all of the preclinical studies in mice that allowed us to develop the pre-IND for this. Then this went into a clinical trial. At that point, it went off the moonshot. Next. And now it's funded through an NCI grant. So this is a good type of graduation where we see that we can fund something, a drug like this that was discovered at MD Anderson, all the way through preclinical studies into clinical trials. Next. You might have to click several times. One more time, I think. Thank you. So this is another example where we had partial success and partial failure. So we started a personalized vaccine trial in advanced pancreatic cancer, where we were able to get tissues from metastatic sites and do immunopeptidome analysis and sequencing on them and identify potential neoantigens and tumor-associated antigens that we then made into peptide vaccine complexes, about 5 to 10 peptides per vaccine, and start a clinical trial in advanced cancer. We realized about 2.5 years into this that this was not working. Patients were not responding. The vaccine was probably being used too late in the disease process. And so we terminated the trial and that was off the moonshot. So again, if something is not working, we need to take stock and say, this is not working, we need to move on. You can't keep funding somebody for 5 years just because they are in the moonshot. But something good came out of all of that, which is we did identify several shared antigens that were found not only in pancreas cancer, but in other diseases. Some of these we were able to clone the TCRs, the T cell receptors, and make products. And so part of this now has been licensed to a company. There have been additional NCI funding for some of this work. So something good came out of all of this. It was not completely a failure at all. In fact, something really good came out of this in the sense that we were able to license this product, and there's clinical trials now open based on one of these products, the VGLL1 right there, you see. And so again, part of this is to have a very clear-eyed vision in terms of progress and make decisions of pulling a project or pivoting it to another, realigning it to another direction based on how it's going. Next slide, please. And the third model, if you can click one more. Thank you. The third model, which is we call the hybrid model. So again, the first model was we support all of the pre-IND studies, and then it goes into a clinical trial. The second model is we support all the preclinical studies and then it's licensed by a company. And the third model is we partner with a company and we fund part of the work that's done inside by the moonshot, and then part of the clinical trial costs, etc., which are the more expensive costs, are done by the company. So this is an example where a company called Iovance that makes T-cell therapies was very interested in how we were able to grow T-cells out of pancreatic cancer tissue. So not just primary tissues, but small metastatic cores. We had a success rate of over 85% in growing these T-cells, including CD8 enriched T-cells, not just CD4 T-cells, but CD8 enriched T-cells. And this was all work that was funded by the moonshot. Iovance got so interested in this work that initially they were not running a clinical trial in pancreas, but they actually started a clinical trial in pancreas based on this data from our moonshot. And they partnered with us where they were paying all of the clinical trial costs, and we were doing all the fun science, like doing single cell sequencing on the T cells, figuring out how the T cells were behaving in vivo and how they change in culture. So all of this cool stuff, all the science that we call expanded correlatives, that was being done by us, funded through the moonshot. And actually this paper, the entire sort of single cell sequencing and trajectory data on probably the world's largest collection of TILs from pancreatic cancer patients, that manuscript has just been written and it should be submitted in the next couple of weeks. So we're very excited about that. And this is another example of how an internal philanthropic effort like the moonshot can partner with a company and sort of really make progress in this disease. Next slide, please. I wanted to show this one example. This is a clinical trial that's ongoing at MD Anderson of delivering siRNA against KRAS. Again, RAS, as I said, is very important, and now there are a lot of RAS drugs that are in the clinic from companies like Amgen and Mirati, and others are coming. But this is a genetic way of targeting RAS with siRNAs that are packaged inside exosomes and then delivered to patients. And all of the preclinical work on this, including the Nature paper in 2017, the GMP production in-house that was done from mesenchymal stem cell derived exosomes, all of that was done and supported through the moonshot. And through the moonshot now, this is amazing. This is all supported through a moonshot funds at MD Anderson. We have now opened a clinical trial. We've now dosed 5 patients with these MSC-derived exosomes, and this trial is continuing. It's now a phase 1 trial in dose escalation. If you just go to the next slide. I always show this slide as an example of what it takes to do team science, where you take something that's in the lab, like an iExosome with KRAS siRNA, that was used to treat mice, and the amount of work that goes into getting it into patients is tremendous. And it took a lot of patience, a lot of funding. We persisted with this because we believed in this particular therapy. And so we'll see what happens in patients, but just to show, it took us 4 years, but 83 colleagues across 17 departments, but we did it. We took an asset that was built at MD Anderson, tested in mice, did the GMP production, did all of the IND enabling studies, and went all the way to the FDA and got that approval to perform this phase 1 trial, entirely funded by our moonshot effort. So I think this is the second last slide. I think I have one more slide. Yes, so this is my summary slide, and I hope I'm on time here, which is 20 years of team science. What lessons have I learned? First of all, pancreatic cancer is a hard challenge. Working in teams is a must. Don't underestimate the importance of teams. Single individuals can't do this. Second, focus on top 2 or 3 priorities. The funding is not limitless. Any foundation has a certain amount of money. You got to figure out what are the 2 things you're going to fund it for. Models are wonderful, but ultimately science has to happen in patients. Be facile about changing track. As I said, sometimes we abandon projects. We change investigators, we realign projects. Don't be shy about doing that. Rigorous external peer review and accountability. Every year, 3 times a year, I go before our SAB and present our moonshot progress, and they grill me like anything, but that's the only way that we can retain the accountability in terms of the work we have proposed. you have to involve junior investigators. Everything in our Stand Up to Cancer, moonshots, in all of the consortia grants that I have been PI on, we always have junior investigators because senior figureheads alone will not work. They're often very busy. The folks who are actually going to make things happen are the junior folks. So you have to involve them in these team science efforts, generously share credit and resources. And again, this is the last point. I say this because a very famous scientist here told me this when I asked him, what should I do when I got my first large team science grant? And he said, make sure you get a very good program manager. That's the most important thing. So invest in program management. Don't just use all the money to fund the science. You need somebody who's going to keep all the scientists on track, make sure they're getting the work done and milestones are being met. So program management is a huge part of team science. Next, please. And thank you very much. I really appreciate the opportunity to speak with everyone.
Fabrice, are you there? Okay. Okay, so sorry, I didn't know I had to actually. Thank you very much for this very exciting talk and summary of your thought along the years in the US. Maybe I can start with one question. You mentioned the prospective cohort on diabetes. Do you have already some insights regarding the role and the information given by following hyperglycemia in patients?
Yeah, so we have recruited about 1,000 patients, and we have 4 pancreatic cancer patients already, and 3. So it doesn't sound like a lot, but remember it's less than 1%, and so the total in 10,000 we expect to find about 100 patients. So we have 4 patients already, and the trial has just started. So, and of those 4, 3 were actually diagnosed at early stage, so they were resectable, which is unusual. Again, it's only 4 patients, so I don't know how this is going to all pan out, but it's happening, and so we'll figure out whether we are actually able to see a stage shift in the diagnosis of patients or not. So.
Okay. And we have, of course, very much interested by all the strategies that are developed for early detection, because as you said in your introduction, it's probably one of the best way to progress. What about the detection of early lesions by imaging, with the progress of imaging technologies? You mentioned it, but you did not do any comment.
Absolutely. No, I completely agree with you. Again, I had to pick and choose. I can speak for the whole day, but I had to pick and choose a 45-minute talk. So I didn't talk about imaging. Imaging is very important. And in fact, in our Stand Up to Cancer grant that we have received, we have a full aim on imaging and using radiomics and sort of cinematic rendition, so to speak, of 3D rendition of images in the pancreas. I have beautiful images that I can show someday if I have time, but I didn't have time today. Imaging is very important. Molecular imaging, computational approaches. This is why one of the key things, and I think I mentioned this when we were speaking, it's not enough to just have cancer biologists in these teams. You have to have physicists. You have to have folks who are doing molecular imaging because they are going to have to work on developing these techniques. A key part of this when we talk about early detection is we talk about biomarkers, whether it's circulating tumor DNA or proteins or microRNAs. But suppose you find these abnormal biomarkers, the next question is, where is it coming from? Where is the lesion in the pancreas? And by definition, if you're doing early detection, these are going to be small, tiny lesions that may be missed by conventional CT scan or MRI. So you can't do biomarker work without doing the concomitant imaging development that will allow you to pick these tiny lesions. So that's very important, critical.
Great. We have a question by Nuria Malatz from Spain, I guess.
Hi, Nuria, how are you?
No, yeah.
Fine. Thank you, Anirban. Thank you very much for your talk. It has been highly inspiring. So I loved your points on prevention, although you gave a very comprehensive view, and actually you showed the paths to follow, no? In a way, also in Europe. So this is not a question to you, but a point to suggest to ARC, and I would like ARC also to join and combine force with other funding agencies in Europe in order to actually to enhance the efforts in Europe altogether on pancreatic cancer, and then also to join to Anirban to huge and beautiful projects that you are conducting. So this is just a suggestion. Thank you very much again, and nice meeting you.
Thank you for those kind words. And I do think that it's extremely important in a disease like pancreas that, you know, obviously many basic science discoveries are going to come from single investigators, and those are critical. Those discoveries are important. But I think we need to do kind of take a page out of how the pediatric oncologists have done it, how for leukemias, they all banded together and formed these oncology groups. It's the same thing with pancreas. If we can come together and bring all the institutions together, then there is so much strength in numbers. Instead of 5 patients, we'll have 500, and we'll learn so much more if we can do it that way. So I'm a big believer in that.
I have a question that I already raised this morning, but what is your current understanding of the early step of pancreas cancer development? We know now that you have a lot of cells with oncogenic mutations in normal tissues, but we need some changes at the level of the tissue itself to get these cells becoming tumors. Yes. What do you know about these steps in the pancreas itself, specifically?
Yeah, that's a great question. And so I think it's important to think of this in a couple of different ways. Number one, the traditional thought used to be, well, you just need mutations to have cancer. But now we know, as you exactly said, in tissues like the esophagus, skin, endometrium, there are tissues that have many mutations, huge patches of Notch1 mutations, etc., but they never become cancer. And so clearly you need things more than just mutations to become cancer. And I think, for example, you need transcription factors that alter the transcriptome. You need alterations in the epigenome, something permissive that will change the entire sort of differentiation status of the cell and make it into something more amenable to neoplastic transformation. So just the mutation is not going to be enough. You also need transcription factor alterations. You need epigenomic alterations. So they all have to sort of happen together to create sort of what we call oncogenic competency. The other thing I want to emphasize is we always think of this as the cancer cell because, traditionally we are used to doing tissue culture in dishes and it's just the cancer cells, but in the body they don't exist in isolation. And now we know from mouse model and work from many scientists that there is incredible and early interplay and crosstalk between the tumor cells and the microenvironment. So as the tumor cells are sort of becoming more neoplastic, they are sending out signals to the fibroblasts and to the immune cells, telling them to behave accordingly. For example, the immune cells allow then immune surveillance to be bypassed, which allows the cancer cells to survive, right? The fibroblasts in turn send signals back to the cancer cells or nutritional elements to the cancer cells that allow them to survive in that tremendous hypoxic, nutrient-deprived microenvironment. So there is continuous crosstalk that's happening, and these changes are happening very early. They're not happening late in the game, they're happening very early. And we know now from studies done in precursor lesions, single cell analysis, and spatial transcriptomics that the immune contexture and the fibroblast architecture changes very early in these cases, in humans, not just in mice, in patients too, suggesting once again that the conversation, the crosstalk between the tumor cells or even the precancer cells and the microenvironment is very early on. So again, just to emphasize, Mutations are not enough. RAS is probably the earliest change that happens. It's critically important, but RAS alone will not give you pancreatic cancer. As you well know, RAS mutations are found in patients with pancreatitis. Many elderly individuals will have RAS mutations in their pancreas, but that doesn't give you cancer. You need transcription factor alterations, you need epigenomic changes. That combined with the microenvironment is what will create the total milieu for these cells to become cancer.
Great. Well, I think we have no other questions. So thanks again. It was great to have you on board today. Very useful presentation and discussion. Thanks a lot.
My pleasure. Thank you very much.